to yet another video. I'm your host for the session today. My name is Neelam Mehra, and today I have come up with the topic, the tricks that you can use to revise Unit 2 faster. As we know that the exam season is going on, and CSIR NET 2022 exam is not very far away. So it is very important for the students to utilize these last few days smartly. So with the same agenda in mind, I'm here to help you out. Today I'll be sharing with you the top 10 tips and tricks that you can utilize to revise the Unit 2 syllabus of CSIR NET syllabus. So there will be free sample papers also that you can download from the link given in the description below. As we know that Unit 2 basically deals with the subject cellular organization. And this subject, the topics that are involved in this subject are very less as compared to the other units. Whole of this subject is majorly based on your memorization skill. How effectively you can grasp a particular concept and memorize it during the time of exam, it will basically decide how good you can score in this unit. So, analyzing the previous year question papers, I have come up with some of the very major important topics that are frequently being asked during CSIR NET syllabus from this unit. So, first topic is membrane structure and function. So in this topic, you need to be thorough and study in detail about the structure and function of membrane. What is the composition of membrane? What are different types of phospholipids and proteins that are involved, and how they are asymmetrically located within the membrane. Then comes lipid raft. It is a very important topic that you need to be thorough with. Another important topic which you'll be dealing with would be the cholesterol, which will decide the fluidity of the membrane and the asymmetry of the membrane. Knowing the concept about cholesterol and its distribution within the membrane is very important. Another important topic is protein transport pathways to various organelles. Yes, so here you need to be thorough with the protein trafficking and how the proteins are being transported from ER lumen. The secretory proteins are majorly being transported from ER lumen, where it is being produced, to the outside the membrane, and how the membranes which are being produced in cytoskeleton are being allocated to various organelles. All these topics you need to be thorough with. Third major important topic of this unit is cytoskeleton. So in this, you need to study in detail about what are different types, what are different components of cytoskeleton, such as actin filament, microtubules and intermediate filaments. You need to understand what is the difference between these type of filaments. You can use comparative table to understand that. Then comes what are different types of proteins, such as kinesin and dynein, which are involved in this unit, how they are responsible for carrying the cargo in which direction, and what are their functions in cellular processes. So all these things you need to be thorough in this particular topic of the unit. The fourth important topic is about structure of chromatin and chromosomes. This topic is related to the Unit 3 of CSIR NET syllabus, that is molecular biology. So if you need to have good hold on Unit 3, you need to study this topic thoroughly. So here the concepts that you will be dealing with would be, there will be basically statement based question that would be coming in part C, where you will be asked about chromatin structures, what are chromatin chromosome packaging, and transposons also come here. Another major important topic which you'd be dealing with in this subunit would be about operons. You need to be thorough with different type of operons, such as lac-operon, tryptophan operon, arabinose operons. What are the different components of these operons, and what are the different regulation of this operon takes place with the absence and presence of their substrates. Next important topic is about meiosis and mitosis. Cell cycle steps and checkpoints. This is a very important topic that you need to study. And this topic is very much related to the Unit 4 topic, cancer biology and apoptosis. So in CSIR net syllabus, most of the questions from cell cycle is being combined with cell biology and apoptosis. So what you can do is you can revise or study both of these units or both of these topics from different units simultaneously. In this topic, you need to be thorough with what are different kind of oncogenes that are involved, what are different type of proto-oncogenes involved, and tumor suppressor genes, what are their roles in cell cycle regulation, their names. Another important concept which you'll be dealing in this particular subunit would be about how the cell cycle progression take place, what are different types of checkpoints that are involved in the regulation of the cell cycle, and what are different types of CDKs and cyclins that are important, that are involved, and what are their specific roles in cell cycle regulation. Last important topic is microbial growth kinetics. This is a very short topic, and most of the questions which come from this particular subunit are numerical based. So you need to be thorough with the equations that are involved so that you know at the time of solving the question or giving the exam what equation to use where. Okay, then comes the study plan. You need to have a very good study plan for revising Unit 2 syllabus. As we know that Unit 2 is a very short unit. Only 5 of the subunits are involved in this particular unit. So accordingly, you can prepare a timetable for yourself. In 5 days only, you can revise whole of the cellular organization syllabus. First day, you can give time. You can give 3 hours to membrane structure and functions. Second day, you can give 3 hours to protein transport pathways to various organelles, cytoskeleton. Third day, you need to give 2 hours to structure of chromatin and chromosomes and transposons also. This is a very short topic, so 2 hours would be enough for this. Fourth day, you can give 2 hours to mitosis and meiosis, cell cycle steps and checkpoints. Fifth day, you need to give 2 hours to solving various types of numericals which comes from the topic microbial growth kinetics. In this way, you can make a study plan for yourself. You can revise topics easily and effectively within a short period of time. Then comes memorizing the characteristics of various pumps, transports and inhibitors, what are their roles and how they are involved in diseases. These pumps and transporters, about them you can learn through Koncept wheel, which would be quite handy for you. I'll explain about how to use Koncept wheel to memorize these topics in the next slide. Then comes important topic, RBC membrane components such as band 3. Most of the questions, it is being noticed that frequently repeats in CSIR net paper comes from the band 3 integral protein which is involved in RBC membrane. So RBC has a 2D structure, RBC membrane has a 2D structure which consists of cytoskeleton and lipid bilayer. Lipid bilayer composition basically consists of different types of phospholipids, sphingolipids, cholesterols and integral proteins. In integral proteins, there are 2 types of proteins which are involved here. That are, first one is the band 3 and second one is the glycophorin. So band 3 integral protein which is involved, it basically acts as a transporter, anion transporter. So you need to be thorough with these kind of integral proteins involved. The questions from this topic majorly comes in match the following type, and it is a very scoring type of question. So you shouldn't miss this topic. Then comes, how can we use the concept wheel to memorize different topics in which we need to remember examples, the concepts and functions. So here I have taken an example. First one is secondary active transport. With examples, it is being explained in this concept wheel. Second is the ATP powered pumps. So what happens here is, in concept wheel, the particular topic is being given in the middle, and around that the wheels basically explain about the concepts about the particular topic and the examples that are involved. So this is very handy. You can utilize this to memorize various types of pumps, various types of inhibitors, various types of transporters and ion channels. Then there would be certain topics which would be coming in subunits where you'll be dealing with protein transport in different organelles. It can be studied with the help of diagrams. Diagram-based study of these kind of topics would be very helpful. Here, basically, your visual part of the brain will play a role. So figure-based questions are being asked from this subunit, and it would be easy to solve if you remember these topics with the help of diagrams. So here I have taken two examples. Firstly is the protein transport is being taking place from cytosol to chloroplast. And here the protein is being transported from cytosol to mitochondria. So as we know that both chloroplast and mitochondria consist of two membranes, the outer membrane and the inner membrane. What are different types of complexes or transporters that are involved in both of these organelles? The outer membrane for chloroplast, there would be TOC complex, there would be TIC complex. What are different types of chaperones that are involved? Mostly the chaperone which is involved in guiding a particular protein to the organelle would be HSP70 protein. It will basically guide the protein where it is to be allocated, and it will also prevent the protein misfolding. Yes, so these things you can revise through diagrams. It will be very helpful. Then comes using comparative tables or concept tables to study topics like mitosis and meiosis, skeleton, cytoskeleton types and functions. So here I have taken two examples of concept tables. In first one, we are dealing with microtubules. There are different type of, we are dealing with cytoskeleton. There are different types of cytoskeleton filaments that we need to be aware of. That is, actin filament would be there, microtubule filament would be there, intermediate filaments would be there. So we can make comparative tables to differentiate between these types of filaments, their functions and structures. Second comparative table is about mitosis and meiosis. So here what is being shown is there are different types of events which are taking place in various stages of mitosis and meiosis. So making comparative tables, you can easily study these kind of topics. Yes, in cell cycle control, names of the proteins and their functions have to be memorized. You can use tables for the revision. So I have taken here two tables where in first table, the general name of the proteins are being given which help in regulation of CDK and cyclins. Majorly, two types of proteins are involved in the regulation of CDKs, that are protein kinases and protein phosphatase. What are their functions? It is being described here. Then comes CDK inhibitory proteins. All the types of CDK inhibitory proteins is being given here and it is being explained here. You need to revise them and remember them, and it would be easy for you to do it if you make a table. The second table basically shows the ubiquitin ligases and their activities and the gene regulatory proteins. All the types of names of the proteins are given here and the description is being mentioned here. In this way, you can revise all the names of the proteins and their functions with the help of table. Then comes revising Unit 2 along with the common topics in Unit 4. As we know that the subunit D of Unit 2 syllabus, it basically deals with cell cycle and regulation, and this topic is in relation with the cancer and the apoptosis topic of Unit 4. So what you can do is you can revise both of these topics or study both of these topics together. It will save your time, and the questions that come in Part C in CSIR NET exam basically comes from the questions which comes from the cell cycle regulation topic of CSIR NET exam. It is being combined with the cancer biology and apoptosis. So both of these topics should be studied simultaneously. Okay. Then there will be certain techniques that would be involved that will be studying in cellular organization subject or Unit 2. That would be FRAP, technique called FRAP would be there, and various questions comes in every year in CSIR NET exam from this technique. The microbial growth curve would be there, questions comes from this also. So studying these kind of topics would be easier if you do it through graphs. So I have taken here 2 examples. Here I have mentioned a question which has come in one of the CSIR NET paper, which basically deals with the FRAP experiment. So it would be in the form of graph. It would be easy to solve these kind of previous year question papers that are based on topics such as FRAP, because graphical questions only comes here. If you solve the previous year questions, you can get an idea that how you solve these type of FRAP related questions. Then comes the microbial growth curve. So it is very important that you memorize this curve. This is a hyperbolic curve. You need to know which kind of events take place in which kind of phase, such as what is majorly going on in lag phase, what is going on in exponential or log phase, what is going on in stationary phase and death phase. All these concepts you need to study, and in the form of graphs, these kind of topics would be very helpful. Then comes memorizing the formulas to solve the numerical questions asked from microbial growth kinetics. Practice one question for different type of numericals. So the last subunit which deals with microbial growth kinetics, there will be different types of questions that would be involved. You need to memorize each and every equation and solve the questions which have come in previous year question paper. So first equation that you'll be dealing here would be how to calculate CFU, that is colony forming unit. The equation for this is number of cells multiplied by dilution factor divided by plating volume. Second equation that you'll be dealing with would be mean growth constant, where mean growth constant is being represented by small k. It is basically number of generations per unit time, where small n is basically number of generations. This value can also be calculated through the formula called log nt minus log n naught divided by 0.31 multiplied by t, where 0.30 is the value of log 2. Another equation that you will be frequently using would be nt equals to n naught multiplied by 2 to the power small n, where nt is the number of cells at time t which would be asked in the question. You will be given the value of initial cell number, and you will be given the value of small n, that is number of generations. Putting these values in the equation, you can easily calculate NT. Then comes experimental based questions. There will be certain questions which will be totally based on experiments. So to revise these kind of topics, what you can use is you can revise them by question and answer method. Practice the experiment based questions from previous year papers. So these kind of topics, there is no direct theory for the answers to be explained. What you can do is, for these kind of topics, you can solve the previous year questions and get a good explanation for the particular answer and understand these type of questions, because there is no theory based concept. So here I have taken an example where it is basically being asked that in order to study the intracellular trafficking of protein A, it was tagged with GFP, that is the green fluorescent protein. A complex is being made where A is being combined with GFP. Using fluorescence microscopy, it showed that A and GFP complex, it co-localizes with LAMP1. In the presence of baphyllomycin A, which is an inhibitor of H plus ATPase, which is a V-type ATPase pump, A and GFP complex does not co-localized with LAMP1. Instead, it co-localized with LC3. So, which statement would be true here? In these type of questions, as we know, there is no theory for this to explain. You can only use the previous year question papers for the explanation of these type of questions. So here the options are being given. AGFP targets the ER in the absence of baphyllomycin A, which is wrong. Second is the autophagy is required for trafficking of AGFP to lysosome. Third is baphyllomycin A facilitates targeting of AGFP to ER. Fourth is baphyllomycin A facilitates targeting of AGFP to the mitochondria. I'll give you 3 minutes to solve it in your mind. Then I'll tell you the correct answer. Yes. So, if we know the concept of protein trafficking, we know that LC3 is a marker which is present during the autophagolysis in lysosome. So, second option would be correct. I hope all of you would have marked this right answer. Then there would be certain tips that you need to keep handy in mind before solving the questions in exam. Firstly, attempt the strong section. In strong sections such as your strong section could be section B or section C. In your strong section also, solve the questions from the prepared topics initially. Then attempt the rest of the sections later. Solve the questions you are confident about to avoid the negative marking. Only those questions you are confident about, just solve those questions and leave the rest of the questions so that you can avoid negative marking. Be careful of part C, as it looks tempting since there is 4 marks you can get directly from one question, but you can also lose 1 marks if you mark incorrect answer. So be very careful while you solve the part C of this unit. I'm totally sure that all these tips and tricks, if you utilize while revising cellular organization or unit 2 of CSIR net syllabus, you will score high in this particular unit. Thank you.